finishing up chapter 11, focusing now more on the action potentials and how neurons communicate. All right, so this is a nice summary figure of neuron communication, and then we'll be going through them more in steps. So first of all, the generation of the action potential that starts at that axon hillock, where the axon begins. And essentially what it's going to do is it's going to ultimately open up voltage-gated sodium channels. They'll allow it to be propagated from one node of Ranvier to the next, and it just kind of jumps along until it gets to the end, to this axon terminal. Then it's going to open up voltage-gated calcium channels. As calcium rushes in, these synaptic vesicles containing specific neurotransmitters are going to join with that membrane, and those neurotransmitters are going to be released into the synapse here, which is the space between the neuron and what it's binding to. Then those neurotransmitters are going to interact with ligand-gated voltage or ligand-gated channels on the postsynaptic cell to then cause a response in those. So first of all, talking before we get into action potentials about graded potentials. And what happens here is a graded potential, as this is showing here, so following the numbers 1, so something is causing sodium to come into the cell. Again, that's going to cause a depolarization, but then if another stimulus occurs, now we get a little bigger depolarization. Then if we get another influx of sodium for some reason, now we get a little higher. And ultimately, we get to the summation point to where if we get two events occurring at the same time, they join together because there's not enough time for sodium-potassium pumps to reestablish the resting membrane potential. So if we get these two happening right beside each other, now we get a larger influx of sodium that can ultimately lead to a change and opening of those voltage-gated channels. And this is just summarizing kind of the steps of those graded potentials, just another way of kind of explaining that previous figure. So now if we get into the action potentials, again, this is the main way that neurons communicate. So the graded potentials summate. So the influx is they're receiving signals from the dendrites. Those graded potentials help to actually start a action potential, which is at what's called the trigger zone. So if they reach a specific threshold and we get that depolarization, then it's an all or nothing principle. So it has to reach this threshold, but after it does, it's the same, it's the stimulus, it's just as strong as a stimulus, whether it was a small or a greater influx of sodium or stimulus. So once we reach that threshold, then the action potential is the same every time. So we have the 4 steps we'll go through next, which is depolarization, repolarization after potential, and then the return to rest. So first of all, looking at all of them together. So again, we have this graded potential. We're getting a change in the membrane potential. Once we reach whatever this threshold is, then we always get the same depolarization every single time. It doesn't matter how big of the graded potential we get. If it reaches threshold, we get the line in red every time. So we get this depolarization where it becomes more positive inside than negative. Then it's repolarized back to negative, and then our after potential ultimately returns us to rest. All right, so starting first with our resting membrane potential, we've already talked about, but at rest, voltage-gated sodium channels are closed. Also, there's an inactivation gate. We'll come back to that. It's open. So they have the ability to open, but currently there's no membrane potential to activate them. Voltage-gated potassium channels are closed. So none of our voltage-gated sodium or potassium channels are open at this point. So the outside of the membrane is more positive compared to the inside. So the inside is negative, the outside is positive. And that's what we talked about. Again, more potassium and proteins on the inside, more sodium and chlorine on the outside. All right, now whenever we get to the depolarization, so if we reach that threshold, sodium activations or sodium channels open. Whenever they open, potassium channels also open, but they're much slower. So depolarization happens when the inward sodium, because remember sodium is high outside, it's also positively charged. So it really wants to come in where there's not a lot of sodium and the inside of the cell is more negative. So there's a huge driving force for sodium to come in. So as it does, that's going to make the membrane potential reverse. It's going to become more positive, usually around 35 or so millivolts, but that number varies. So that resulting depolarization opens additional voltage-gated sodium channels that are close by. So more sodium comes in and we get more depolarization. That then opens more voltage-gated sodium channels, and you can see how this continues. So this is an example of positive feedback to where, again, opening of one triggers the opening of additional ones. Right, then the repolarization. So once it gets to a certain point, once it reaches that maximum depolarization, that inactivation gate that I'd mentioned. So they have a voltage gate on the sodium channel that opens and closes based on membrane potential. But also whenever the membrane potential reverses, there's a gate called the inactivation gate. And that goes over top of those sodium channels. And even if there's a membrane potential, they can't open. So it blocks them into a position where sodium cannot move through them. Now, all other potassium channels, voltage-gated potassium channels open. Again, they started opening in the depolarization too, but they're just slower. And so once the sodiums are inactivated, that allows the potassium to have more of an effect. So the sodium movement into the cell stops and potassium is going to move out. Now there's a huge force for it to go out because there's a high concentration inside of the cell, so it wants to go out. And now the inside is more positive and the outside, and now at this point the outside is more negative. So now that positive potassium ion really wants to go out because it's going with its concentration gradient, also with that charge or voltage gradient. All right, now we get to the end of repolarization. So our activation gate, so the gate on the voltage gated, it closes because now the membrane potential is back to negative inside, positive outside, and that inactivation gate is going to open. So this would then allow sodium to then move back in. So that reestablishes the conditions for the sodium channels. All right, then we actually have an after potential to where it's going to dip further down. It's going to become hyperpolarized because of those voltage-gated potassiums. They're going to stay open a little bit longer than what it takes to bring the membrane potential back. So those sodium-potassium pumps are still going to be working, but extra potassium is going to leave the cell. So it's going to make it even more negative on the outside, hyperpolarizing it. Okay, then the resting membrane potential gets restored after the voltage-gated potassium channels close, and then the sodium-potassium pump reestablishes. So at this point, it's more negative inside, more positive outside. But before we get back to rest, we need to put the sodium back outside and the potassium back inside. And that's the job of that sodium-potassium pump that moves those 3 sodium outside, brings 3 potassium inside, or 2 potassium inside. There's also a point here that's referred to as a refractory period, and there's an absolute and a relative refractory. And this is the point where we have a little bit of change in sensitivity. So an absolute refractory, it's completely insensitive to another stimulus. So even if there was a graded potential that would start an action potential during this absolute refractory period, sorry, I circled the wrong one there. So during that absolute refractory period here, kind of in purple, it doesn't matter if another stimulus is coming in that would reach threshold. We're already in an action potential, so it can't go any higher. So there's no way to reactivate an action potential. Then during the relative, so whenever it becomes hyperpolarized, it's a relative refractory because since it's more negative than it is at the beginning, again, hyperpolarized, it would take a much stronger threshold, a much stronger stimulus than it would when the membrane is at rest. So again, if we're down to negative 90, now much more sodium has to rush in before that threshold could be reached. So this is that relative refractory. So again, during this period, it's really hard for another action potential to be generated. So we have a few different frequencies here. So we have a sub-threshold stimulus. So this does not cause the graded potential to initiate action potential. So there's an influx, there's a change in the membrane potential, but it doesn't reach threshold, so no action potential is generated. The threshold is what's needed in order to initiate the action potential. Then on the other side, we have submaximal stimulus. And this is everything between threshold and the maximal stimulus. Maximal, as the name implies, is the maximum frequency of an action potential they're firing. And this is based on that refractory period, how quickly it can reach and reestablish to set off another action potential. So the fastest it could possibly get reset and another one fire off is the maximal stimulus. Then we have a supramax stimulus, and this is any stimulus stronger than the max. And these cannot produce a greater frequency though. Again, because of the refractory, it can only go so far. You may be activating it, but it can't possibly work any faster. And this is just showing what we're talking about here. So again, subthreshold. So there is a change in the membrane potential, but it doesn't reach the threshold. So no action potential occurs. Again, here's our threshold stimulus. Now we're going to get an action potential. We're going to get one of those, and then it's going to come back to rest. Submaximal would maybe be a couple. So we're going to get that action potential, but again, it can't go any faster. It might increase the frequency a little bit, kind of increases the duration here of that action potential. Then if we increase the frequency even more, now we're at the submax. So again, both of these here being submaximal, so they're increasing the frequency. It's more than just one action potential. But again, we're not quite maxed out. Then at maximal, so this part of the neuron cannot generate any more than 4 action potentials in this given amount of time. And again, it may not be that number, but the frequency, they cannot possibly fire action potentials any faster than what they are doing. And then even if a stronger stimulus comes in, again, we're still at the max, because that's what max means. We can't go any faster. The stimulus may be stronger, but the result's not going to be any different. We get the number of action potentials that we get, and that's based on that refractory period. Even though it's being stimulated, it can't generate another action potential any faster. All right, and this is just kind of writing all that out. So again, the duration of that absolute refractory period determines the maximum frequency. Again, how quickly can it reset to that resting membrane potential to make it excitable again? So the stronger stimulus will result in greater frequency of action potentials, but the magnitude's the same. So it still changes and becomes depolarized at the same amount. Doesn't make it any more depolarized. But it does make the action potential happen faster than it would. Okay, so again, all or nothing. It happens or it doesn't. So the magnitude doesn't change, but the frequency can. So the target of the action potential interprets a higher frequency. Again, more and more and more action potentials coming faster and faster. That's interpreted as a stronger stimulus. For example, so it would produce a stronger, say, muscle contraction or more secretion from a gland than what a lower frequency would. And this is just talking through all the steps of that action potential. Again, I'm not going over them again, but it's just a nice summary to kind of hit all those steps. All right, now once the action potential reaches that axon terminal, we get close to that synapse, which is the junction between two cells. And this is where the action potential in one cell causes some type of action in another. So there are two synapses. We have the presynaptic, that's the one transmitting the signal, and the postsynaptic is the one receiving the signal. And we have electrical synapses and chemical synapses. We'll mainly focus on the chemical. So electrical synapses are mainly, we only see these in cardiac muscle and in most of our smooth muscle, but not in our nervous system. And essentially what this is where an action potential in one cell actually produces a local current of sodium that moves into an adjacent cell and activates an action potential in the adjacent cell. So it's a way of moving the action potential from one cell to another using gap junctions. So those gaps that link two cells, well, as sodium comes into one cell, it can also go into another. So that action potential moves from one cell to the next through those gap junctions. And those are referred to as electrical synapse because we're still using the change in membrane potential caused by the influx of sodium. Now in neurons, we're talking more about chemical synapses. So again, we have our presynaptic terminal. That's the end of the axon that is releasing the neurotransmitter, the signal, into the synapse. And that gap is called the synaptic cleft, just a space between the pre and the postsynaptic cells. And then the postsynaptic membrane is the membrane on the neuron it's talking to, or it could be a muscle cell or a gland. And then neurotransmitters are the chemicals that are released due to that action potential. So we have what are called synaptic vesicles. They're going to fuse, as I mentioned at the beginning, when calcium comes in to the axon terminal, it causes those vesicles containing neurotransmitters to be released. And then they diffuse across the synapse to that postsynaptic membrane. And the neurotransmitters bind to receptors like ligand-gated ion channels. And then the opening of the channel would then cause an effect. So if we look at the chemical stimulus, again, this was kind of our overall, but if we look closer here at the synapse, again, we have our presynaptic terminal. It has these synaptic vesicles in here. What's going to happen is when the action potential comes down, now whenever the membrane potential changes, this is going to open voltage-gated calcium channels. So now calcium is going to rush into this axon terminal, and that causes these synaptic vesicles to fuse or join with the membrane, and it releases those neurotransmitters into the synapse. Then the neurotransmitters are going to bind to ligand-gated ion channels. And the binding of them would open up the ion and allow the ion to come into the postsynaptic membrane. So what happens once the neurotransmitter is released? Well, it very quickly needs to be removed so that the cell knows whether to keep responding or to end the response. So the method of removal depends on the neurotransmitter. So we're not going to go through every neurotransmitter. Acetylcholine is shown here as an example. But the neurotransmitter, again, the effect needs to be short-lived, so they're rapidly destroyed or removed. And there are three ways to get rid of them. Breaking down the enzyme, so this is showing an enzyme here that's a release, and it would break down acetylcholine in this case, or a neurotransmitter to make it inactive. We also have reuptake. So in this case, we have reuptake transporters. And that allows that acetylcholine to go back into the presynaptic cell. Okay, so again, it enters this presynaptic terminal, and then it just forms more of that neurotransmitter. It gets packaged back into a vesicle, and then ultimately can get released again. So it's a way of recycling the neurotransmitters. And then the fourth way is just it can diffuse away. So it just moves out of the synapse and around other cells. So it gets diffused away from it. And so some neurotransmitters go through all three of these. They all go through diffusion, but some have reuptake, some don't, some have enzymes, some don't. Again, just understand the general ways of removing them. All right, so again, just our summary here, back looking at this again. Here's our dendrite. So in this case, we have a cell that is releasing a ligand. That ligand is going to bind, opens up ligand-gated sodium channels, so we get depolarization here. That depolarization is enough to come to our axon hillock and open up those voltage-gated sodium channels. That's going to let sodium come in, and they're going to allow that action potential. So we get our depolarization, then our potassium channels are going to open to get repolarization, and that keeps moving all the way down until we get to the axon terminal. When that action potential reaches there, calcium floods in through those voltage-gated calcium channels, causes the synaptic vesicles to join with the membrane and release those neurotransmitters onto our postsynaptic cell. Again, just another way here, this is just walking through that more in text form, if that's more beneficial than what I just described. All right, so these neurotransmitters can be classified based on their chemical structure. Monoamines like dopamine and serotonin, they're structurally very similar, but their effect on the postsynaptic, like they're excitatory or inhibitory, or just what they're doing, their mechanism of action. So they're causing secretion of a hormone or muscle contraction, something like that. So they can have excitatory or inhibitory effects. So they can either cause the target cell to be excited or inhibited, and we'll come back to this in a little bit. They can also have an inotropic effect, so ion here, meaning that they bind to ion channels, like ligand-gated ion channels. Other times, though, a neurotransmitter, instead of binding to an ion channel, binds to a different receptor, a G protein coupled receptor is an example. But this is going to change the effect on the cell. It's going to cause the cell to do something like release a gland, or it may be a gland that's releasing a certain hormone. So it's going to cause binding of the ligand to these receptors, causes production of that hormone, and then release of the hormone from the gland. So the function is determined by the receptor on the target cell. Now the next couple slides are just some examples, some tables of neurotransmitters. You are not responsible for knowing all the neurotransmitters and what they do. I'm just including these so that you get an idea of the variety that we see. So acetylcholine, for example, it's an inotropic, meaning it binds to a ligand-gated ion channel. And so then that allows an ion to move through, which excites or inhibits that cell. Serotonin and so dopamine, both of these are mood stimulators. So again, they're regulating mood. That's a more metabotropic effect. Again, dopamine, for example, cocaine releases lots of dopamine and it blocks the reuptake. So it stays in, the dopamine stays in the synapse longer. So it makes us feel better, has a happier mood. That's a metabotropic effect. We have some that are more inhibitory, like GABA and glycine. So they inhibit cells. They make it harder for an action potential to occur. Okay, so some other ones, even like nitric oxide here. This is what is used in things that cause vasodilation. So nitroglycerin that helps prevent a heart attack. It releases and causes the production of nitric oxide that acts as a neurotransmitter. Endocannabinoids, so think about marijuana and the effects that it has. It activates these endocannabinoid, resembles them and binds to those receptors. So then what effect do they have on the postsynaptic membrane potential? So we have EPSP, which is excitatory postsynaptic potential. This means that depolarization occurs in response. So again, a ligand-gated ion channel for sodium. So when the ligand binds, sodium comes in, and that causes depolarization. That would be an example of excitatory postsynaptic potential. Whereas an inhibitory, IPSP, this would cause hyperpolarization. So in this case, a neurotransmitter, say, binds to a ligand-gated ion channel, but it lets chlorine come into the cell or potassium to leave the cell, which would then make it more negative on the outside. So it makes it more hyperpolarized. So it's less likely that an action potential would occur. So it's more inhibitory. And then finally, we have neuromodulation. So neuromodulators, these influence the likelihood of an action potential. So they're, again, making it a little easier or harder in order for that action potential to occur. We also have axonic synapse. Those are the ones where the axon of one neuron synapses with the presynaptic terminal of another. So these happen a lot in our central nervous system. We have presynaptic inhibition. So this is reduction of a neurotransmitter released. So for example, endorphins can inhibit pain sensation. So endorphins are released, they're going to modulate, they're going to make it harder for that action potential to occur. And then we have presynaptic facilitation. So now the amount of neurotransmitter released in the terminal increases. So more of it is being released, so a greater response is going to occur. Then our spatial and temporal summation. So a single postsynaptic potential is not sufficient to reach threshold. So it's many postsynaptic potentials combined, so we call that summation. All that information is taken into that trigger zone at the axon hillock, and there are two types, spatial and temporal. So in spatial, this is where the action potential of 1 and 2 are in two different dendrites. Okay, so we have our cell body here, we have a dendrite coming off over here, and another one over here. Well, both of these are going to depolarize the cell, but they're not close together. It's on two different parts of the cell, two different dendrites, but ultimately they're depolarizing the cell so that trigger zone is activated. Whereas in temporal summation, these action potentials arrive close in succession, so they're coming right beside each other, and that's going to activate and allow enough change in the membrane potential that we get activation at that trigger zone. Okay, and then finally, our neurons all have to figure out what to do with all this. So an action potential at the trigger zone, when we have those graded potentials, is a result of both excitatory and inhibitory summation. So at any dendrite, there may be some axon terminals that are releasing inhibitory neurotransmitters, others that are releasing excitatory. So the cell body has to take in all of that information. And so by the time it reaches that trigger zone, it either reaches threshold or it doesn't. So are the excitatory and inhibitory canceling each other out? Is there more excitatory, more inhibition? All of that gets combined or summed together to ultimately either cause an action potential or not.